Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today, we're going to be covering Chapter 7 in our MCAT Biochemistry series, and this chapter is titled RNA and the Genetic Code. In this chapter, we're going to cover the following objectives. First, we're going to discuss the genetic code itself, including the three main types of RNA, how codons work, and how mutations can affect protein synthesis. Then we're going to walk through transcription, how RNA is made from a DNA template and the processing that happens afterward. After that, we're going to move on to translation, where we're going to learn how proteins are built from mRNA at the ribosome and what happens after translation is complete. Next, we'll explore how gene expression is regulated in prokaryotes through operons, inducible systems, and repressible systems. Finally, we're going to finish by looking at how gene expression is controlled in eukaryotes. We're going to cover transcription factors, gene amplification, and chromatin remodeling. With this introduction in place, let's jump into the first objective, the genetic code. Both DNA and RNA, they share the same language. They both use sequences of nitrogenous bases to store and transmit genetic information. These bases act like letters in a biological alphabet. And just like letters form words, base sequences form codons that can be interpreted and translated. Now, a gene is essentially a specific unit of DNA that contains instructions to build a particular protein or RNA molecule. In other words, it's a coded message, and that message can be expressed by going through two essential steps, transcription and translation. And that leads us into one of the most important frameworks in biology, which is the central dogma of molecular biology. The central dogma describes the flow of genetic information in cells. It tells us that information is stored in DNA, it's transcribed into RNA, and then it's translated into a protein. And that's the basic direction of gene expression. Now, this is very much big picture, but we're going to unpack each step of this process throughout the chapter. We're going to start with the types of RNA and how the genetic code works before we dive into transcription and translation in detail. With that being said, let us discuss the different types of RNA. There are three major types of RNA that play key roles in gene expression. First, we have messenger RNA, or mRNA. Next, we have transfer RNA, or tRNA. And finally, we have ribosomal RNA, or rRNA. Let's go through each one, starting with messenger RNA. mRNA carries the genetic instructions from DNA to the ribosome where proteins are made. It's created during transcription when RNA polymerase reads the DNA template and then synthesizes a complementary RNA strand. Each set of three bases on an mRNA strand is called a codon, and each codon corresponds to a specific amino acid. These codons are read during translation to determine the sequence of amino acids in a protein. Now, in eukaryotes, each mRNA strand usually encodes for just one protein. This is called monocistronic message, mono meaning one. In prokaryotes, though, a single mRNA can contain multiple coding regions for several proteins, and this is called polycistronic message, poly meaning many. Next, we have transfer RNA, or tRNA. tRNA is essential for translating the genetic code into the language of proteins, and its job is to bring the correct amino acid to the ribosome during protein synthesis. tRNA has a distinct cloverleaf shape when it's drawn in 2 dimensions, but it folds into a more compact shape in 3 dimensions. At one end, it has an anticodon, which is a set of 3 bases that are complementary to a codon on the messenger RNA. At the opposite end, it carries an amino acid. When the anticodon on the tRNA pairs with a codon on the mRNA, the ribosome knows which amino acid to add to the growing chain. So let's role play here. You're a codon on the messenger RNA strand. Let's say you're AUG. You're lonely, you're just waiting in line, hoping to meet your perfect tRNA match. Then comes along your big, beautiful, so muscular tRNA soulmate. His anticodon is UAC, and he's everything you've been waiting for, and he pairs perfectly with you. And to top it all off, he brings you a gift. No, it's not flowers. It's even better. It's an amino acid that matches your codon, your dream date. Cute. Now, a tRNA, it's considered charged or activated when it's attached to a specific amino acid. This process is catalyzed by enzymes called amino-acyl tRNA synthetase, which ensures that each tRNA is paired with the correct amino acid. We're going to get to that shortly, like what each codon refers to in regards to amino acids, but keep that in mind, all right? Each codon refers to a specific amino acid, and tRNA brings that amino acid when its anticodon matches the codon on the mRNA. And this step is essential for translation because if the wrong amino acid is attached, then the protein is going to be made incorrectly. Finally, we have ribosomal RNA, or rRNA. This is a structural and enzymatic component of the ribosome. It helps position the messenger RNA and the transfer RNA correctly during translation, but rRNA isn't just scaffolding, it also has enzymatic activity. In fact, the ribosome's active site, which is the part that forms peptide bonds between amino acids, it's catalyzed by the rRNA itself. Because of this, ribosomal RNA is classified as a ribozyme, which is an RNA molecule that acts like an enzyme. That was a major discovery in molecular biology because it showed that RNA isn't just a passive messenger, but it can have catalytic functions too. So together, messenger RNA, transfer RNA, and ribosomal RNA, they form the core machinery that allows cells to read genetic information and build proteins. Now that we understand the roles of the different types of RNA, let's really talk about how the information in messenger RNA is actually read and translated into a protein. Again, if you think of a gene sequence as a sentence, then the basic unit of that sentence is a 3-letter word called a codon. A codon is a sequence of 3 nitrogenous bases on the messenger RNA strand, and each codon is going to correspond to one specific amino acid. So going back to our role-playing example, the codon AUG, it codes for the amino acid methionine. When you look at the codon chart that's shown on this page, we read the codon by looking at the first letter on the left, the second letter across the top, and then the third letter down the side. So if you're looking up the codon AUG, you'd start with the row for A, you'll follow the column for U, and then you go down to the row where the third base is G. And that points you to the correct amino acid for that codon. Now let's connect this to translation. During translation, each codon in the messenger RNA, that's going to be matched to a complementary sequence on a transfer RNA molecule. That complementary 3-base sequence is called the anticodon. So for example, if the codon on mRNA is AUG, then the anticodon on the tRNA, that's going to be UAC. And that tRNA carries the correct amino acid and delivers it to the ribosome. From here, there are 2 very important types of codons I want to highlight. The first is the start codon. The codon AUG signals the beginning of translation. It not only starts the process, but it also codes for the amino acid methionine, which is always the first amino acid in a newly synthesized protein. Then we have stop codons. These are codons that do not code for any amino acid. Instead, they signal the ribosome to stop translation and release the completed protein. There are 3 stop codons you need to know: UAA, UGA, and UAG. And a common way to remember these is with a little mnemonic. UAA stands for you are annoying, UGA is you go away, and UAG is you are gone. Coincidentally, these are also my morning affirmations, so very easy to remember. Now to summarize, codons are how genetic information is read in triplets. Each codon brings in a specific amino acid, and the sequence of codons determines the final protein. The process starts with AUG and it ends with a stop codon. Now, there are a few more important features of the genetic code that we need to cover. Let's start with the idea that the genetic code is degenerate. This means that more than one codon can specify the same amino acid. For example, there are 6 different codons that all code for the amino acid leucine. And this redundancy, it provides a built-in buffer against mutation. So if a change occurs in the sequence, it actually might still produce the same amino acid and the protein would remain unchanged. A big part of this protection comes from something called the wobble position. The wobble position refers to the 3rd base of a codon, and this base is more flexible than the first 2. A mutation at the 3rd position often doesn't change the amino acid at all, and these types of changes are called silent mutations because they don't alter the final protein. On that note, let's actually take a closer look at mutations in general and how they affect proteins. The first category we have are point mutations. A point mutation is a change in just one nucleotide, and depending on where it occurs and what it changes, it can lead to different outcomes. Again, if the mutation occurs in the wobble position and it doesn't change the amino acid, we call that a silent mutation. The sequence is different, but the protein stays exactly the same. If the mutation changes the codon to one that codes for a different amino acid, that's called a missense mutation. And this results in a single amino acid substitution. Sometimes that change is minor, and other times it can really disrupt the protein's function. One classic example of this is sickle cell disease, where a glutamate is replaced by a valine, and that results in the misfolding of hemoglobin. Another type of point mutation is a nonsense mutation. This is when a codon is changed into a stop codon, and that causes translation to end too early. So this usually results in a shortened and or non-functional protein. So far, all of these examples involve a single base being substituted. But what happens when a base is added or removed? This brings us to frame shift mutation. A frame shift occurs when a nucleotide is inserted or deleted, and that's going to shift the reading frame of the codons. Since the ribosome reads the mRNA in groups of 3 bases at a time, even a single insertion or deletion, that's going to completely change how the sequence is being read downstream. And that usually leads to a completely different protein or even an early stop codon. And frame shift mutations are often much more disruptive than point mutations. That wraps up our first objective. We can now move into our second objective, which is all about transcription. Transcription is the process of making an RNA molecule from a DNA template. This is the first step in gene expression, and it's how the cell creates a working copy of a gene that can leave the nucleus and be used to build a protein. To begin, the DNA double helix needs to be opened up, and this job is handled by helicase, which unwinds the DNA and it separates the 2 strands. Now, as this is happening, another enzyme called topoisomerase works ahead of the replication fork, and it relieves tension and it prevents supercoiling. Once the strands are separated, only one of them is used as the template for RNA synthesis. This is called the template strand or the antisense strand. This is the strand that RNA polymerase reads to build the RNA. The other strand, which is not used during transcription, it's called the coding strand or the sense strand. It's going to have the same sequence as the RNA that's produced except it contains thymine instead of uracil. So, how does transcription actually begin? It starts at a specific DNA sequence called the promoter region. This promoter is found upstream of the gene, and it is the site where RNA polymerase binds to begin transcription. Now, before we continue, I do want to take a moment to explain 3 RNA polymerases that are found in eukaryotic cells and distinguish between the 3 because only one of them is involved in the transcription of messenger RNA. RNA polymerase 1, it synthesizes ribosomal RNA, and that happens in the nucleolus. RNA polymerase 2 is the one we're interested in. This is the most important for us now because it synthesizes messenger RNA. It also makes small nuclear RNA, which helps with splicing. We'll get to that later. RNA polymerase 3 synthesizes transfer RNA, and it also produces some ribosomal RNA. Now, going back to transcription, the RNA polymerase is going to bind to the promoter, and a common element within that promoter is the Tata box. This is a DNA sequence that's rich in adenine and thymine, and it helps position RNA polymerase correctly at the start site. But RNA polymerase doesn't act alone. In eukaryotes, it needs help from transcription factors. These are going to be proteins that help RNA polymerase recognize the promoter and bind tightly to the DNA. And they make sure everything is aligned properly before transcription starts. Transcription begins at a defined spot called the +1 site, also known as the transcription start site. From here, RNA polymerase reads the DNA in the 3' to 5' direction while building RNA in the 5' to 3' direction. The product of this is a strand of heterogeneous nuclear RNA. This is the raw, unprocessed form of RNA that's made in the nucleus. But before it can be used as a messenger RNA, it does need to go through several processing steps, which is what we're going to cover next. So once transcription is complete, the RNA we have at this point is not ready to be used for translation, and processing steps are actually going to help it be ready. So before it becomes mature messenger RNA or mRNA, it has to go through a series of post-transcriptional processing steps. These steps are going to occur in the nucleus, and they're going to be essential to making the messenger RNA stable, functional, and pretty much ready to be exported into the cytoplasm for translation. There are 3 major modifications that happen: splicing, 5' capping, and 3' polyadenylation. Let's start with splicing. During transcription, the RNA is going to include both exons and introns. Exons are the coding regions. These are the parts that actually contain instructions for making a protein. Introns, on the other hand, are non-coding regions that need to be removed. Splicing is the process then of cutting out the introns and then joining the exons together to form a continuous coding sequence. This process is carried out by a large molecular machine called the spliceosome. The spliceosome is made of small nuclear RNAs and proteins, and these small nuclear RNAs recognize specific sequences at the intron boundaries, and they help catalyze the removal of said introns. And once splicing is complete, the mRNA now only contains exons. Next, we have the 5' cap. At the very beginning of the RNA molecule, a special molecule called 7-methylguanosine is added to the 5' end, and this modification is called the 5' cap. The 5' cap plays 2 important roles. So first, it helps the ribosome recognize the messenger RNA during translation, and second, it protects the RNA from being degraded by enzymes while it's still inside the cell. The last major modification is the 3' poly A tail. At the 3' end of the RNA, a long stretch of adenine nucleotides is added, and this is called the poly A tail. The addition of the tail is catalyzed by a special enzyme, and it involves adding about 100 to 215 adenines to the end of the transcript. Now, the poly A tail also helps protect the RNA from degradation, and it's going to assist with the export of the messenger RNA from the nucleus into the cytoplasm. So to summarize, splicing removes introns and connects exons. The 5' cap helps with ribosome recognition and stability, and the 3' tail protects the RNA and it helps it leave the nucleus. Now, there's one more important concept to mention here, and that is alternative splicing. Alternative splicing is a process where a single heterogeneous nuclear RNA transcript can be spliced in different ways to produce multiple versions of messenger RNA. That means that from one gene, you can get different proteins depending on which exons are included or skipped. And this allows for protein diversity without needing additional genes. It's super powerful in complex organisms like humans because it allows us to maximize the information that's encoded in our genome. Now we can move into objective 3 and focus on translation. Translation happens in the cytoplasm and it takes place at the ribosome. This process requires 4 main components: the messenger RNA transcript that carries the genetic code, that's 1. 2, the transfer RNA molecules to bring in the correct amino acids. 3, the amino acids themselves as building blocks. And finally, 4, GTP as a source of energy to power the process. Now, the ribosome is the structure where all of this happens, and it is made of ribosomal RNA and proteins, and it has 2 subunits, a large subunit and a small subunit. Now, there is a difference in ribosome size between prokaryotes and eukaryotes. In prokaryotes, the ribosome is referred to as 70S, which is made up of a 50S large subunit and a 30S small subunit. In eukaryotes, the ribosome is larger. It's called the 80S ribosome, and it's made up of a 60S large subunit and a 40S small subunit. Now, even though the math doesn't add up here, these S values are based on sedimentation rates, not direct addition, so they reflect shape and density rather than just size. Within the ribosome, there are 3 key sites where transfer RNA molecules interact with the messenger RNA. First, we have the A site. This is where the incoming tRNA carrying a new amino acid first enters the ribosome. And the A site stands for aminoacyl tRNA site. Next is the P site. This stands for peptidyl site. This is where the growing polypeptide chain is held. The amino acid from the A site, that's going to be joined to the chain held in the P site. Finally, we have the E site or the exit site. So once a tRNA has delivered its amino acid, it moves to the E site and it exits the ribosome. Now, we're going to make more sense of this as we go through the steps of translation, which include initiation, elongation, and termination. Let's start with initiation. During initiation, the small ribosomal subunit binds to the messenger RNA. The exact way this happens really determines and depends on whether you're a prokaryotic cell or in a eukaryotic cell. In prokaryotes, this small subunit is going to bind to a specific sequence on the messenger RNA that's called Shine-Delgarno sequence. This is a ribosome binding site that's located just upstream of the start codon, and it's going to help position the ribosome correctly on the messenger RNA. In eukaryotes, the small subunit is going to bind to the 5' cap of the messenger RNA. And from there, it can scan along the messenger RNA until it finds the start codon, which is always AUG. Once the start codon is located, an important molecule called the initiator tRNA binds to it. This tRNA carries the amino acid methionine, which is always the first amino acid in a newly synthesized protein. The initiator tRNA is going to bind directly to the P site of the ribosome here for this first step, and that's unique because all the other tRNAs, they're actually going to enter at the A site. Now, at this stage, a group of proteins called initiation factors are also involved. What do? They help guide the ribosome to the messenger RNA They help stabilize that interaction, and they make sure that everything is assembled correctly. Once the initiator tRNA is in place and the start codon is recognized, the large ribosomal subunit goes ahead and joins to complete the full ribosome, and that marks the end of initiation, and the ribosome is now ready for the next phase. The next phase is elongation. During elongation, the ribosome reads the messenger RNA one codon at a time, and from that builds the protein by linking amino acids together. Here it's really worth repeating that the ribosome has 3 sites. The A site is where the new tRNAs carrying amino acids enter. The P site holds the tRNA that is carrying the growing peptide chain. And the E site is where empty tRNAs are released from the ribosome after they've delivered their amino acid. So here's what happens during elongation. A new tRNA enters the A site, and it's carrying the next amino acid that corresponds to the codon in that position. Then, a peptide bond forms between the amino acid in the A site and the growing peptide chain in the P site. And this reaction is catalyzed by the enzymatic function of the ribosome called peptidyltransferase. This is actually part of the ribosomal RNA. After the bond is formed, the ribosome undergoes a process called translocation. So here, it shifts forward along the mRNA in the 5' to 3' direction, and as it moves, the tRNA that was in the P site, it moves to the E site and it exits the ribosome. The tRNA that was in the A site is now carrying the growing peptide chain, and it moves to the P site. And now the A site is open for the next incoming tRNA. In this process, it repeats and repeats for each codon in the messenger RNA. Along the way, elongation factors are going to help guide tRNAs into the A site, and they're going to help coordinate the movement of the ribosome down the messenger RNA. And these elongation factors also use GTP as an energy source to power that process. Eventually, the ribosome reaches a stop codon, and that brings us to the final step, termination. There are 3 stop codons, UAA, UAG, and UGA. These codons actually don't correspond to any amino acid, so there's no tRNA that binds to them. When that stop codon enters the A site, though, translation can no longer continue. Instead, a protein called a release factor binds to that stop codon, and this release factor will trigger the ribosome to cut the bond between the final tRNA and the completed polypeptide chain. That's going to release the finished protein into the cytoplasm. Then additional proteins called termination factors, they can also assist in this step by essentially disassembling the ribosomal subunits and cleanly ending translation. Once the protein is released, the ribosomal subunits fall apart, and the messenger RNA, it can either be translated again or degraded depending on the needs of the cell. One final note here. Many eukaryotic proteins have special regions called signal sequences. These are short peptide tags that direct the protein to its proper location in the cell. We're going to come back to this idea a little bit later, but keep that in mind. Once a protein is made, though, the job isn't quite finished yet. Most proteins actually need to be modified before they can actually become fully functional. And these changes are called post-translational modifications. And they are essential for helping the protein fold correctly, reach the right cellular location, and perform its biological function. Let's break this down. There are 2 broad categories of modifications you should know. The first involves structural changes to the protein itself. So these include cleavage, where part of the protein is cut off, and this can activate the protein or allow it to change shape. The other type of structural change is signal sequence removal, which means trimming off a short peptide that help direct the protein to its proper destination. Or you can also have subunit assembly, where multiple protein chains come together to form a functional complex. Now, the second category of modifications include chemical additions to the protein. So these are covalent modifications that will add specific groups to the protein. Each one has a unique effect. Let's go over 4 important ones that we need to know for the MCAT. The first is phosphorylation. Phosphorylation adds a phosphate group to the protein, and it's often used as a switch to turn proteins on or off. It's one of the most common regulatory mechanisms in the cell, and it plays a huge role in signaling pathways. Next is carboxylation. This adds carboxyl groups, which are really important for calcium binding. So this type of modification, it can help stabilize certain protein structures and interactions, especially in proteins that bind metal ions. Next, we have glycosylation. This adds sugar chains or carbohydrates to the protein. This is going to help guide proteins to the correct destination in the cell, and it can also play a role in immune recognition and cell signaling. Finally, we have prenylation. This adds lipid groups to the protein, which help anchor it into cellular membranes. So proteins that need to associate with membranes, like signaling proteins, they often require this to localize properly. And these modifications, they allow the same protein to be used in different ways or to respond to different cellular conditions. And with that, we have now completed Objective 3, and we can move into Objective 4. In Objective 4, we're going to shift our focus to gene regulation, specifically how prokaryotic cells control gene expression. In prokaryotes, genes that are related in function, they're often grouped together into a unit called an operon. And this concept comes from the Jacob monad model, which was the first major model of gene regulation, and it was proposed in bacteria. Now, an operon, this is going to be a cluster of genes that are all transcribed together under the control of a single promoter. Instead of having one promoter for every gene like we see in eukaryotes, bacteria often group genes with similar functions together and then control them with a shared regulatory system. This makes gene expression more efficient for prokaryotes, and it's especially important in response to environmental changes. Each operon is going to contain 4 important components. The first is the regulator gene. This gene isn't part of the operon itself, but it's located nearby. And what it does is it codes for a protein called a repressor. The repressor protein is going to be responsible for turning the operon off when it's not needed. The second important component is the promoter. This is the sequence of DNA where RNA polymerase binds to start transcription. So it's important for initiating gene expression. Third, we have the operator. This is a region of DNA that's going to be located between the promoter and the structural genes. It's where the repressor protein binds. And when the repressor is attached to the operator, it will physically block RNA polymerase from moving forward, and it will essentially stop transcription. The fourth and final component is structural genes. These are the actual genes that are being transcribed. They usually encode proteins that work together in a common pathway. For example, enzymes needed to break down a sugar. From here, we want to talk about 2 different types of regulatory systems, inducible systems and repressible systems. Let's start with inducible systems. In an inducible system, the default state is off. That means the genes are not expressed unless something specific happens to turn them on. So here's how that works. A repressor protein is normally bound to the operator. That will block RNA polymerase from transcribing the structural genes. So this is an example of negative control, where gene expression is going to be prevented by the presence of a repressor. However, when an inducer molecule is present, it's going to bind to that repressor protein, and that will cause the repressor to change shape and then fall off the operator. Once the repressor is removed, RNA polymerase can now move forward and begin transcription. So the presence of the inducer turns the gene on. And a classic example of this is the lac operon. This is found in E. coli, and it contains genes that allow the cell to break down lactose, which is a type of sugar. The goal, though, is to produce these genes only when they're actually needed. So when there's no lactose in the environment, a protein called the lac repressor, it binds to the operator region of the operon, and that's going to physically block RNA polymerase from moving forward. So transcription can't happen. And that makes sense. There's no need to produce enzymes that break down lactose if lactose isn't even present. But when lactose does become present or available, some of it gets converted into a molecule called allolactose. This acts as an inducer. And what does an inducer do? It's going to bind to the repressor, it's going to cause it to change shape, and then it will fall off. All right? Once the repressor is gone, RNA polymerase can now proceed with transcription, and the cell begins to make the enzymes that are needed to break down lactose. Now, in addition to this negative control through the repressor, the lac operon is also regulated through a positive control mechanism. This involves a protein called catabolite activator protein, or CAP, the CAP protein. This is going to help enhance transcription, but it only becomes active when glucose levels are low. So when glucose levels are low, the concentration of a signaling molecule called cyclic AMP, it increases in the cell. And cyclic AMP binds to CAP, and this complex then binds to a site near the promoter of the lac operon. And the binding of this CAP cyclic AMP complex, it assists RNA polymerase by making it easier for the polymerase to bind and initiate transcription. So to kind of summarize this, the lac operon is inducible. It's normally off, but it turns on in the presence of lactose. And it's controlled in two ways. First, through the negative control by the lac repressor, and second, through positive control by the CAP cyclic AMP complex, which boosts transcription when glucose is low. Now, what about repressible systems? In a repressible system, the default state is actually on. So the genes are usually being transcribed and expressed. However, under certain conditions, the system can be turned off. And here's how that works. So in the absence of a signal, the repressor protein is inactive, so it can't bind to the operator. What that means is that RNA polymerase is free to transcribe the structural genes. But when a specific molecule is present called a co-repressor, it's going to bind to the repressor protein and then activate it. And once it's active, the repressor goes ahead and binds to the operator. And when it's there, it'll block RNA polymerase and it will shut off transcription. A good example of a repressible system is the trip operon, which is responsible for the synthesis of the amino acid tryptophan. That's why it's called TRP operon. Now, when tryptophan levels are low, the repressor is inactive and transcription proceeds and the enzymes for synthesizing tryptophan are made. Great, wonderful. But when there's a lot of tryptophan, it's going to act as the co-repressor. So it will bind to the trip repressor protein, activate it, and then that repressor goes ahead and binds to the operator, and it will block transcription and prevent any further production of tryptophan. So in this case, the presence of the product, aka tryptophan, that turns off the pathway that makes more of it. And this is an example of feedback inhibition, which helps the cell conserve resources. Now we can move into our last and final objective. This objective discusses control of gene expression in eukaryotes. In eukaryotic cells, gene expression is tightly controlled, and that's because not all genes are constantly active. They're only expressed when they're needed. And unlike prokaryotes, where regulation mainly happens at the transcriptional level, regulation in eukaryotes can occur at several different points. These include controlling how accessible the chromatin is, how transcription is initiated, and how much the gene is amplified. Let's start by discussing chromatin structure. Chromatin is the combination of DNA and histone proteins that make up chromosomes, and it can exist in two forms, euchromatin and heterochromatin. Euchromatin is loosely packed and transcriptionally active, and this open structure is going to allow enzymes and transcription machinery to access the DNA. On the other hand, heterochromatin is densely packed and transcriptionally silent. So it's basically shut down to transcription, and it's going to appear dark under the microscope. To regulate gene accessibility, the structure of chromatin can be chemically modified. And this is where histone modification comes in. One major modification is histone acetylation. Enzymes called histone acetyltransferase, or HATs, they add acetyl groups to histone proteins. This loosens the chromatin structure and it makes the DNA more accessible, which will increase transcription. In contrast, histone deacetylase, or HDACs, they remove those acetyl groups. So this is going to tighten the chromatin and it will decrease transcription. Another way gene expression is regulated is through DNA methylation. DNA methyltransferases add methyl groups directly to DNA, and this will typically lead to long-term gene silencing by preventing transcription factors from binding or by recruiting proteins that make the chromatin more compact. From here, we can start to talk about transcription factors. Transcription factors are proteins that are going to help determine which genes get transcribed. And they essentially do this by helping RNA polymerase bind to the DNA. These proteins will search the DNA for specific sequences called promoter and enhancer regions. Promoters are usually found within about 25 base pairs of the transcription start site, while enhancers are typically located further away, so more than 25 base pairs upstream or downstream. Each transcription factor is going to have two recognizable domains. That first domain is the DNA binding domain, which is going to latch onto specific DNA sequences known as response elements. And these are sequences that are going to be near or far from the gene that help regulate it. The second domain is the activation domain. This region allows the transcription factor to interact with other proteins like RNA polymerase or some other co-activators to either activate or suppress transcription. Now, some transcription factors act as activators and they will boost gene expression, while others are going to be repressors that shut down gene expression. When transcription factors bind to enhancer regions, especially those that are kind of far away from the gene, what they can do is they can cause the DNA to sort of loop around so that the enhancer comes close to the promoter. This kind of looping brings all of that necessary machinery together and it allows for efficient transcription to begin. But beyond these mechanisms, eukaryotic cells can also increase gene expression through gene amplification. So that means producing extra copies of a gene to kind of boost its output. And a similar idea is gene duplication, where a gene is copied and the extra version is also expressed. Both of those strategies result in a higher level of gene product. Now, to summarize the big picture, gene regulation in eukaryotes, it happens at multiple levels. It starts with chromatin structure. Acetylation opens DNA, deacetylation closes it, and methylation silences it long term. Then we have transcription factors, which locate promoter and enhancer regions and use their DNA binding and activation domains to really regulate gene expression. And finally, we have amplifying mechanisms like enhancers, gene duplication, and gene amplification to further control how much of a gene product is made. And with that, we've completed everything we need in this chapter. I truly hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.